check the admission. I still need to put a key up for this. I'll try to do that right when I get back. And if you have any questions, just shoot me an email or come see me. And so for our last portion of the class, the final, there's going to be 50 points on the last 4 lectures here. So that'll be just like a regular exam in terms of scope and depth and vocabulary and all that. And then we'll have 100 points that are comprehensive. And if you have not yet looked at the old final, I recommend that you do that because you get a sense of kind of the big picture questions. So for these more comprehensive questions, I'm going to try to pick things that I consider to be mainly highlights that you would take away from the course, including major concepts or the more important genes or the more important kinds of terminology. So I'm probably not going to get into super nitty-gritty, but at the same time, you know, it's sort of inherent that you do ask specific questions, so you know, you need to know some of the specific things too. In terms of studying for the final, what I recommend is to do kind of a funnel-shaped approach where you might look first at the old final exams and then go back to look at this year's exams, hourly exams, and last year's hourly exams that are posted. Because by doing that, you'll go through what's already the kind of cream of the crop material for test-wise. And by the time you've done that, you've gone through already 8 exams plus the final. That's already a pretty good overview of the course. So when you're going through that, if you find major areas, say like spermatogenesis, that it's just gone, make a mark, and then you could go back and review that area in a little bit more detail just to kind of refresh your memory on those things. And then if you still have time beyond that, you could go back to the lectures and sort of say, what are the major highlights for this lecture? What are the concepts? And I've actually listed the concepts in your study guides. So you could perhaps use that as a tool to just say, can I articulate what this concept is all about? And if the answer is yes, I think you're probably in pretty good shape. So I would recommend something like that as opposed to just getting out the notes for lecture 1 and plowing through all those and keep on plowing through the notes for all 35 lectures. So, okay, let's go ahead and look at some of these lectures in detail now. So the first one was about sex determination in mammals, lecture 31, I believe it is. And questions on this process here?
No, it's on 28.
Oh, yeah, 28. What was 28? Endgerm?
There's a first one.
Oh, yeah, okay. Frontal arches and endgerm. So yeah, nothing super complicated here. There's just a number of different structures that are derived from endgerm, and most of them have to do with some sort of connection along the gut tube. You'll see some of the same paradigms coming up that we talked about before, like reciprocal induction. So a lot of interaction between the endoderm tissues and the surrounding mesoderm tissues signaling back and forth to make organs. You can find an example of a master regulatory gene in terms of the PDX1 transcription factor for pancreas development. What else is highlights from this lecture? Regional gene expression. So along the anterior posterior axis, you get different sets of unique transcription factors being turned on, and this helps establish the different territories. Okay, so I guess we're looking pretty good in that one. Let's go ahead to lecture 31 then with sex determination in mammals. So you could pretend that you were me and you had to write an exam question for some of this material on here. There are things that you could ask about the testes determining factor, the SRY gene, some of the move it or block it types of experiments, and what that ended up being, if it was changing males into females or females into males, or what the scenario there was. Let's see what I put down for concepts here. So then we also had the primary and secondary sexual differentiation. So you should have an understanding of what the difference is between those two. What does it mean to say that sex determination is chromosomally determined? And have a look at that model, the slide with the model on there about male versus female development and the genes that are involved. And you know, the main genes I'll be interested in are just the SRY and the SOX9. But in principle, the take-home there is that both the male development and the female development are active in gene-directed processes. And so it does trigger on whether or not SRY is present, but you'll either shine into one pathway or into a different pathway depending on if it's expressed or non-expressed. But the point is, you do need pathways in both cases to make male or female development. Yeah, question?
Do you expect us to know like the differences between the exos system and the ZW system?
So the question is about knowing the differences between the XY system for mammals and the ZW for birds. Yeah, I guess I would say yes to that. It's not, just be aware that there are different ways of doing it out there, and that these are still actually chromosomally determined, right? It's just different chromosomes, and it happens that in birds it's switched as to, it happens to be the female with the two different chromosomes. Yeah?
There was a question on the practice final about the Wolfian duck, which sex does it persist in? And the answer just says male only. And I was reading online, and the definition for Wolfian duck said that it persists in females, but it's vestigial. So is that wrong, or?
Well, vestigial. Sorry.
You're going to degenerate? That's what our slide should be.
Yeah. So it may depend on the exact species, but this word vestigial means that it's very, very reduced and basically non-functional. So technically there might be like a little bit of a remnant of it that is still there after it degenerates. But as a primary, as a major structure within the gonad, it goes off. Alrighty. So this gives you, kind of a format, a paradigm for how you could instigate a sex within a mammal. And then to contrast with that, we also looked at sex determination in Drosophila. And so there's some interesting kind of differences here, because Drosophila also has chromosomally determined sex, and it also has an XY system. So it's similar to mammals in that way. But it's different in the way that the cells actually evaluate what their genotype is. So instead of referencing a Y chromosome with an SRY, they ignore the Y, and they just evaluate this X to autosomal sex ratio. So have a look at that material in the next lecture, and make sure that it's clear how this is determined in Drosophila. Any questions on lecture 32 with the non-mammalian sex determination for Drosophila?
The splicing part, the sex-dependent splicing, we're not going to have to know like where, so what's missing, what develops. Just know that there's different sections cut out that creates different.
Yeah, so there's one slide in there in the Drosophila lecture that I've parsed out into 4 different slides, and then I gave them a whole.
Yeah.
So try to say, what's the take-home paradigms here? So I won't ask you something really detailed like, which exon is spliced out in males or something like that, but the idea that you could get differential splicing in males versus females. The idea that it's the females that makes a sex-lethal protein. Both males and females are initially making RNAs, or at least pre-mRNAs. Then they alternatively splice them, and because of that event, only the females makes a functional sex-lethal protein. So that's part of the mechanism, the details of the mechanism for Drosophila. And, so that applies to sex lethal, it applies to some other genes that are also in the pathway, like transporter, and, what's the other one, not shovel, but double sex, Okay, so also in lecture 32, besides this mechanism of Drosophila sex determination, we talked about dosage compensation. So that's another biggie from that part. And you may want to be familiar with how different organisms accomplish that goal. So there's 3 different ways to do it, but you get to the same endpoint in each case. And in mammals, we do that by inactivating one of the X's and making a body out of it. Then we also talked a little bit about the cases where sex determination is not chromosomally determined. And it may rely on environmental factors such as temperature. And somehow, though, you still have to get that environmental information to play into gene expression. It's the same thing for plants and these meristem identity genes. In this case, we have to get environmental information to play into the neuroendocrine system so that you can see some differences in expression leading to hormone differences, testosterone versus estrogen. So we have an enzyme aromatase that's sensitive to temperature, and that wild type job of that enzyme is to convert testosterone into estrogen. So the temperature can influence how much activity is present and then how much estrogen is present or not present in the organism down the line. Any other questions on that lecture? Okay, that would be a good lecture to look at the practice question on the exam for, because that actually asks you to apply some of this information, and sometimes it's kind of one of those things that makes perfect sense when you're just reading it, but then when you have to answer a question, you're like, what? It can be kind of confusing. Okay, so heading on into plant life here, then we had a massive lecture with all kinds of terms and pollen development and egg development. Any questions on that part of the material? Okay, that lecture has, again, a lot of vocabulary, so make sure you know that cold, and that will be a big advantage for that lecture. And then that lecture also, if I were a student, the way I would study that is to make diagrams. Because you have 2 or 3 different processes that are happening here, like you're starting out in the anther, and you have this microspore in the mother cell, and then it's getting converted to a microspore, and then that thing's getting converted to a pollen grain down the line. So see if you can just study that up and then put the book closed and draw that whole process out. And if you can do that, I think you would probably be able to answer really any question I could throw at you on the process. And the same thing for egg development in the female part of the plant. There's all these different terms and all these different cells and kind of a continuum of cell division and relabeling and designating these cells as different structures, eggs or synergids or whatnot. So if you can visualize how that all fits together, it should help. Yeah.
Does the same thing kind of apply to like oogenesis?
Definitely.
Like if we do the diagrams, we should be fine.
Yeah, I think so. So I sometimes, I don't get a lot of people coming to office hours, but sometimes the people I get are the really, really successful students, and they're just trying to lock down the last details. And so I've asked them, what are some of your most successful strategies for studying? And this is one thing that I've heard, is that they like to take, multiple slides in a lecture all on the same topic and see if they can condense it down to a 1-page thing with all the terms labeled, and these people have it color-coded and everything else too. So I think that's great, and that would definitely be a good way to synthesize. So in general, studying things where you make diagrams like this, what you're doing is actually having your brain recall the information, out of the memory and answer some question or make some connection. And that's a much more efficient way of studying than just, say, rereading your notes or just recalling the information by reviewing it. So you want to try to, put yourself in a situation that's similar to the test, where you don't get a look at your notes, you have to just remember it and make connections to a new sort of question or scenario. Okay, and then so this brings us pretty much to, let's say, stomata development, and we had an in-class exercise on that. So have a look at that. I think I posted that online. And you guys looked at the different phenotypes, and you looked at a genetic pathway, you looked at loss of function, gain of function, and so how did it all get out? Yeah.
Will we need to know the specifics of that pathway, or if we're asked questions on it, will we be shown it like we were in
I'll
the
show you.
class
So if I ask a question on that, I'll show you the pathway, and I'll ask you something very similar to what we did on the in-class exercise. Okay. And then we have today's material, which is the maths box. So I generally do have a question on there that says, okay, if the B class is mutant, what phenotype do you predict? So that should be quite easy to answer. You just need to remember the little wild type box diagram and how you get the 4 different kinds of information and what order the worlds are in. And if you know that, I think you'll be able to figure out, on the spot, any mutant phenotype that I would throw at you. Okay, so I'll just open it up now for just any question. I don't know how far people have gotten in terms of studying, but are there things that have come up that you'd like me to go over again, or just questions? Yeah.
I have a question about the reproductive cloning question you have in your final, mainly about the cats. So the question is, if a mother cat was a calico cat, the cloned offspring, a female, would also be calico, but the color and arrangement of fur patches on the offspring will be different than the mother. Name the process that is responsible for the difference in coat pattern. So, is that neural crest migration problems, or?
It could be. So it could be if this calico cat had the mutant background that caused the melanocytes to not migrate as far. So that was going to be something that's a little bit different from one individual to the next within the species, although they're all going to have some expression of that phenotype. However, that's not really, so if you put that, I'd have given you full credit because that's a correct answer, but that's not what I was going for. So did people hear the question, or should I go? Okay. So what is the answer to that?
Exon activation.
Exon activation. So just because exon activation is going to be random and unique for each cell in the early embryo, so that's going to give a uniquely color-colored pattern animal down the line.
Because underneath of it, it says white spotting genes affects neural crest migration.
Right, so that's the alternative answer that probably 2 or 3 people in the class would come up with, and everyone else would have said exon activation. It's kind of a neat question though, I think, because it ties together these 3 different things. Like here we have reproductive cloning, where you're saying genetically this animal is identical to another individual because we used one of its diploid cells to recreate it. And then we have the white spotting, so that tied in the neural crest migration, and then we have exon activation, so that tied in something that we talked about much later in time.
And then would the cat be normal? Like, that's essentially the other 2 questions.
The cat should be normal, yeah. I mean, unless you count possible other neural crest defects, if it does have that. Now, you may find a question or 2 on the old exam that has some material that we didn't cover, but you should recognize that as being something you've never heard of before, and so don't worry about it. Like numerator and denominator genes we didn't talk about this year.
We only talked about the one type of bone formation, right? Just
We
the
didn't
one-
talk about
just-
bone formation either.
Just the facial bone.
Yeah, so there are a couple of lectures that you guys missed out on because you took it in the summer. So I'm sorry to deprive you of some material, but you didn't get to hear about bone formation or kidney formation. We didn't do kidney, yeah. Briefly mentioned Yeah,
So Hox genes always have a homeobox, but they're not the only genes that could have a homeobox. Is that correct?
right, so, Hox genes, so there's the homeobox gene family. These, by definition, all have a homeobox somewhere in the sequence.
Sure.
And then, members of that would include all of the Hox genes.
Okay.
If you're in mammals. It's gonna include some other things if you're in Drosophila, like, ABD was one of them, the 8 genes that are in the HOMC complex. It also contains, in mammals, a few random genes kind of scattered around the genome, like CROX happens to be one that has a homeobox. It's expressed in the brain, but it's not part of the 4 clusters. So the Hox genes themselves are part of these 4 clusters. The HOMC cluster, which is the original cluster in Drosophila, and then these guys are not in clusters, but just here and there.
So the HOMC cluster is only for Drosophila.
The HOMC cluster is just a historical name for this original cluster of 8 in Drosophila, correct?
Okay. So then, and T-box genes don't have anything to do with this, except for the fact that they also have like a conserved sequence, yeah.
Right, so other gene families that we could talk about as having sequences in common among the family members, T-box, paired box or PAX genes,
That's all.
MADS box,
MGF.
the myogenic regulatory factors. These are all beta HLH transcription factors that are involved in muscle development. And I don't know, I can't remember if they actually have a domain in common or not. So yeah, there are a lot of different families that we can identify.
And so I have a question about Wnt and the TGF-beta superfamily. Wnt
Okay.
is not a part of the TGF-beta superfamily, correct?
Correct.
Okay.
So Wnt is its own thing, but,
But the bone morphogenetic proteins, those are all part of the TGF-beta family,
Right.
right?
Diagrams, like for the flower part, where it shows all the steeples, like are we gonna have to like diagram it, or just know which parts are which and what? What's
so
involved in
you
the
would not
development?
have to draw a diagram, but, for purposes of studying, you might want to draw a diagram. Just because that makes you, especially if you draw a diagram without looking. But no, that's way too hard for me to grade, so I need something. We can blaze through so you guys can get your grades if you done.
I don't know if that's a line or.
My thoughts? Okay, so you can feel free to email me, I'll try to keep an eye on things. And I think we're probably in good shape. I've enjoyed having you in classes.
Whenever I read it too in the book, I was like, I told you, I was like, it's mesoderm.